Dr. Lambert with a video on DNA structure. So we're going to look at how DNA is put together here. So remember, DNA is a nucleic acid, specifically is deoxyribonucleic acid. That's what DNA stands for. DNA. Okay, so nucleic acids are one of the types of biomolecules that we talked about. And we said with those biomolecules there were monomers that made up larger biomolecules that were the polymers. So the monomer in DNA is the nucleotides, and the polymer is the nucleic acid. So the subunits, nucleotides, look like this picture that I've drawn here. They will have a sugar. This sugar, for all nucleic acids, will be either ribose or deoxyribose. Now we're talking about DNA, so the one that we'll be seeing in these drawings is deoxyribose. Okay? Either way, as you can see, the sugar is here in the middle of our nucleotide. Our nucleotide in our nucleic acids will always have a phosphate group, and it will also have a base. So in deoxyribonucleic acid monomers, the nucleotides of DNA, the bases that you will see are shown here. A, T, C, G. And what do those stand for? They stand for cytosine, guanine, adenine, and thymine. Now these are ring structures. We'll talk about that in a minute. And the big thing about them is that they contain nitrogen. So they are often referred to as nitrogenous bases. I'll put big letter N there so that you remember that they contain nitrogen. So the polymer is the nucleic acid, and this one here that looks like a twisted ladder is my version of DNA, deoxyribonucleic acid, and I will be referring to it as a ladder-like structure. So we'll often draw it like that, and I'll talk about the rails of the ladder, the rails being the sides, and the rungs of the ladder, the rungs being the part, if it were ladder, the part that you would actually climb. So that's DNA with its two strands, two sides, okay, and then under normal conditions it's going to have this helical twist to it. And then there's RNA, which is another type of nucleic acid that we will talk about in future chapters, but notice it just looks like half of a ladder. So that is a single-stranded nucleic acid, whereas DNA is double-stranded. All right, so let's talk about DNA characteristics. What makes it special? Well, as we've already said, it's made up of nucleotides. Okay. And these nucleotides create they create the ladder that we see. So on a DNA molecule, or within a DNA molecule, is where you will find genes. So genes, those that which codes for all the characteristics that you see in a person is found on DNA. So genes, a gene is, let's make that, gene is a segment of DNA. And as I have said before, our genes are an expression of our proteins. So we use that DNA, remember this always, the DNA is our library, right? And it's found inside of the nucleus. And it is used to code for RNA, which is the recipe, okay, for how to make a protein. And the protein is the expression of the gene. Okay. Maybe it's the gene for how to make a particular membrane protein, or maybe it's a gene for eye color. Okay. Whatever it is, it is found in our segments of our DNA. Okay. So how does DNA code for something as different as a membrane protein or eye color? So what is important there is the base sequence. Okay, so the base sequence, so I just talked about that, A, G, C, T. The order that those go in, okay, is what codes, okay, A, G, C, T codes for the gene, for the protein. Okay, actually the DNA would code for the RNA, okay, which would in turn code for the protein. Okay, and we'll look at how that happens in future chapters as well, but just to see the big picture here, it's important to understand that base sequence is really what makes a gene special. Okay, so the bases are very important, and understanding how they align themselves is important as well. So the bases will pair. And they pair up with each other in a very specific way. A will always pair with T, and G will always pair with C. Move that up a bit. Okay, so A and G, remember these are nitrogenous bases. A and G, they happen to have, when you look at their structure, they have a double ring structure. So they're wider. And we call these purines. The double ring nitrogenous bases are purines. T and C are single ring structures, and they are called pyrimidines. Okay, so the base pairing rule is that a purine always pairs with a pyrimidine. So if we were just to make it really simple and draw, say that I'm just going to draw them as actual circles here. So that's a purine, and it always pairs with a pyrimidine, right? You've got this particular width, right? If we were to put a pyrimidine on this side and a purine on this side, that would be fine. It would always stay the same width. If we accidentally, and it happens, accidentally paired two single rings, what would happen to the width of our DNA? It would change, right? Or if we put two purines next to each other, I didn't make those wide enough to really exaggerate what would happen there. Let's make them go like that. Now our DNA is too wide. Okay, so this pairing of a purine and a pyrimidine is what keeps the DNA a uniform diameter. I should say DNA is. Let's say DNA has a uniform diameter. Okay. All right, so the bases are pairing A to T, G to C, purine to pyrimidine, so that the DNA always has a uniform diameter. So the other characteristics that we just have to mention, and we'll see these demonstrated in the next page here, we always have to remember that each, as I said, and I just want to write it down so that you see it, each DNA molecule has 2 strands. It's double-stranded DNA. Okay, so as I draw it like a ladder often, it'll look like that. Okay. So those are the each side, each one is a DNA molecule. Okay, and the bases are here in the middle. That's where you find the bases. The bases are, let me pull this down here like this, make that a little neater so we can read it. The bases are in the middle, and the sides of the ladder, the rails of the ladder, are the sugar phosphate bonds. Okay, so it'll as you look down the rails, will be sugar phosphate, sugar phosphate, sugar phosphate, and then the rungs of the ladder, as I said, are the bases. Okay. Now these are 2 separate strands, but they are held together, and they are held together by, that's how I always show these guys in class, right? Hydrogen bonds. And I'm not really doing it justice because sometimes there's 2 hydrogen bonds, sometimes there's 3 hydrogen bonds, depending on which pair you're looking at here. So I'm just showing you the red dots to remind you that it is hydrogen bonds that hold the 2 strands together. Let's go write that down. Make that number 6. The H bonds are going to hold the strands together. Don't forget that in reality it doesn't actually look like a nice straight ladder. It will twist so that it forms a helix. And because there are 2 strands, you'll hear it referred to as a double helix. And last but not least, and we're going to see how this is demonstrated in the next slide, the strands are oriented in an anti-parallel fashion. And what that means is that they run parallel to each other, just like this ladder, but they're in an opposite orientation. So the one is opposite of the other. So we'll see what that means in the next page. Okay, so let's take a closer look at DNA. And you can see that I've drawn, I've made some of my little nucleotides here, and I've got my nucleotides over here on the side that I can copy and paste and put over here and build DNA. So that's what I'm going to do so you can see how this all comes together. Okay, so I've started my strand here with 2 of the bases opposite each other and ones that parallel each other. And I've got a cytosine and a guanine. C and G go together, they pair, right? So notice how on this C I've got him oriented what I'll call right side up, okay? And then the G, the guanine nucleotide, is upside down. Okay? So these are parallel to each other but opposite in orientation. Now there are 2 carbons that you absolutely have to know by number in the sugar. So remember, the sugar is my green. I'm going to zoom in on him and label it. Okay. So the carbons are numbered, a little bit bigger. The carbons are numbered here. Remember, every bend here is a carbon, and you start here, 1, 2, 3, 4, and then there's one here. I didn't do justice showing that there's one there that is going to attach to the phosphate. Okay, remember there's a little part that comes off of our sugars there. So, who are the important carbons? Of course, they're all important, but the ones that we have to know by number here, number 3 and number 5. And the sugar is the central component, so we say it's the these are the prime carbons. Okay, so we will refer to this carbon right here as the 3 prime, and it looks like an apostrophe, 3 prime carbon, and we'll refer to this carbon as the 5 prime carbon. So you can see in my little picture right here, if we could say upside or downside here, the 5 prime is up and the 3 prime is down. Okay, so if we go put that back over there, let's take a look at this guy. Okay, so on him, we're gonna start from the same spot, so we have to start by the base. So there's 1, 2, 3, 4, and the 5 is right there attached to the phosphate. Okay, so we have to find our important guys here. Number 3, number 5. So this is our 3 prime carbon, and this is our 5 prime carbon. Okay, so on that one you can see that the 3 prime carbon is up and the 5 prime carbon is down. So you can see that these are oriented in an opposite direction. So let's build some DNA with my little molecules here. So I added some nucleotides to my growing strand of DNA. And I'm not doing any particular order at all, I'm just kind of putting them in there randomly. But I want to show you how these connect to each other. So if I zoom in, I'm going to erase this here a little bit. Okay, the 3 prime carbon here is going to attach to the phosphate. So we're going to keep going in this direction, just like that. Okay, so you can see on this side that we have a 5 prime carbon as the, I'll say the up carbon. Okay, so 5 prime on that side, and that at the bottom of our sugar, and the bottom of our DNA strand here, our short little strand, we've got a 3 prime carbon. So we say that this side is the 5 prime to 3 prime strand. It runs 5 prime to 3 prime. So now we're going to complement this strand with the other side. So you can see I've already complemented this strand. I've already complemented this strand here with a C pairing with a G. Okay, cytosine pairing with guanine. So what I want you to do is just on a piece of paper, or pause this and think about it. Think about what this growing strand is going to look like. And I'm going to go ahead and move those on. So if you need to, you should pause it now, because I'm going to go ahead and do that. Alright, so I complemented the strand, but now I have to make the bonds between my sugars and phosphates. So I'm going to go ahead and draw those in. So 3 prime, like that. Okay, and look here, we've got our 3 prime carbon up, right there. 3 prime carbon is up. And And as we travel all the way down here, here's our 5' carbon would have been right here. So the 5' carbon is down. So we say that this side is 3' to 5'. Okay, so now remember with these 5' to 3', 3' to 5', I'm using up and down as my orientation because that's how I drew my picture. But you have to look at the strand as the way however the artist drew it and be able to determine which is the 5' to 3' and which is the 3' to 5'. So it really doesn't have to do with up or down or left or right. It has to do with the orientation of the carbons or of the sugar, and therefore which carbons are leading the strand and ending the strand. Okay, so make sure you're able to see that in different orientations. All right, last but not least, to finish my DNA strand, I want to make sure and add in some hydrogen bonds connecting these guys. Make sure we remember that that is happening as well. These are our hydrogen bonds, H bonds. Okay, so that's it for DNA structure. If you have any questions, please feel free to contact me.